So do you remember on one of the beginning recordings we talked about how the nervous system is made up of neurons and glial cells? And there's a couple of different types of glia cells. Astrocytes, they are named because they looked kind of star-shaped. And they work to synchronize the firing rates of neurons. They also help remove waste materials, including dead neurons. It's not good when neurons die. We tend to keep neurons throughout our lifetime, unlike skin cells or hair cells or blood cells that are replenished. Neurons, we do have stem cells that can make new neurons, but generally there's not a turnover, except for maybe in our taste buds, those sensory receptors, because of burns and stuff, can be regenerated. But generally the neurons that we have are with us for life. Astrocytes can help pass chemicals between neurons and the blood. Microglia, because they look really tiny, remove waste, viruses, little fungi, other microorganisms. Oligodendrocytes produce myelin sheaths in the central nervous system. Schwann cells are myelin sheaths for the peripheral nervous system, and we'll talk about central and peripheral nervous systems a few chapters from now. But here's an example of how an astrocyte, the pink, giant pink one, hooking up with the other neurons to help synchronize the firing rates. And here's some more images. Here's an astrocyte on a capillary, a tiny blood vessel, moving materials or waste. And here's an actual image. Here's a Schwann cell that covers axons to be myelin. Here's an oligodendrocyte that looks a little bit like an astrocyte, but not as with as many sort of limbs. And you see how it's actually covering a couple of axons in this case. Remember, this is for the central nervous system, whereas the Schwann cells in the peripheral nervous system. Microglia, tiny. And then radial glia have a very important role in the developing brain, developing nervous system, because they make a pathway, and the migrating neuron sort of pulls itself along it. When you watch the video on the baby's brain, you'll see radial glia and the migrating neurons. There's some captured video of that occurring, and it's very exciting. Well, if you're like me, you find it very exciting. Schwann cells again, and I wanted to try and show them in their development. This kind of jumps a little bit to chapter 4, but I think you'll enjoy it.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Rampier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Rampier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.